Lori Passmore moved from Canada to London in the 1990s to do a PhD with David Barford at the Institute of Cancer Research. For her postdoc, she moved to Venki Ramakrishnan's lab at the LMB, where she worked on ribosome structure, before starting her own group in 2009. Lori's lab works on the molecular machines that regulate messenger RNA poly-A tail formation, a prime example of the long-term curiosity-driven research that the LMB is so good at supporting and encouraging. With former LMB group leader KJ Patel, she also studies the molecular mechanisms underlying Fanconi anemia. Lori is an EMBO member and is also one of 4 female early career group leaders at the LMB who have won the Suffrage Science Award, which celebrates women in science for their scientific achievements and for their ability to inspire others. Lori, thank you very much for coming to talk to us today. I'd like to start at the beginning of you arriving at the LMB. So you didn't actually apply to Venki, is that correct?
No. When I did my PhD, I was working on this really difficult problem, the anaphase promoting complex. And it was really difficult in those days to get information from cryo-EM because it was still a relatively low resolution technique. We didn't have the tools that we have now. So I decided that in order to really become an expert, I needed to come to the LMB to learn cryo-EM, which is where many of the methods had been pioneered. So I wrote to Richard Henderson. He was the director at the time, and he said, no, he didn't have space for me in his lab. But a few days later, I got an email from Venki saying Richard had passed my email onto him, and he was looking for somebody to do cryo-EM in his lab.
Mm-hmm. And Venki's lab were not a cryo-EM lab at that point?
They weren't. They were doing, everyone in the lab was doing crystallography, and they were all working on the bacterial ribosome. But at the time, Venki really wanted to get into the eukaryotic ribosome and felt that the best way to approach that would be using cryo-EM. So I happily said I would work on the eukaryotic ribosome with Venki, but I asked Richard if he would be my co-advisor because I really wanted to have that EM expertise available to me.
Yeah, because otherwise it would have done the exact opposite of what you wanted, I guess, going into a lab where you were, well, I guess you were viewed as the expert in Venki's lab.
Well, that was the funny thing, is that I felt I was coming here to learn from the experts, and then they hired me as the expert in cryo-EM. There were really relatively few of us doing cryo-EM at that time, probably only around 7 or 8 of us.
Really?
So it was a pretty small community. I mean, at the LMB.
Yeah. Huh. So was it a bit strange coming in as a new person and being told you were an expert?
Yeah, absolutely. I think I always remember the first time I went on the microscope, and Xiaoxia Chen, who's still the manager of the facility, took me down to the EM facility to train me and get me set up on the microscope, and she spent about half an hour with me and said, "You're good to go." And I said, "No, you can't leave me now." And she said, "Yes, I can. You know exactly what you're doing." And so I think then I had to develop that confidence to do it all on my own and actually to teach others how to do it as well.
All right.
Yeah.
Wow.
Yeah.
Was it very different here to the Institute of Cancer Research?
I don't know if it was different. Maybe I was just, I guess it was a completely different environment. I mean, I was viewed as an expert. Another example is, I had worked on this really big E3 ubiquitin ligase as a PhD student. And so KJ Patel used to come and find me in the canteen and talk about his favorite E3 ligase, which is the Fanconi anemia core complex, and ask me lots of questions about E3 ligases and say, wouldn't it be really cool if we could do the same types of studies you did on the APC on the FA core complex? And, at that point, I was just a starting postdoc. But when I started my lab then, several years later, KJ and I hired a postdoc to start working on that, and that then became a big project in the lab.
So you got your feet under the table quite early from the sound of it then?
I guess so.
Yeah. So what were you doing in Venki's lab in terms of project?
So we were trying to understand the initiation of translation. So, how the small ribosomal subunit binds the mRNA and finds the start codon and begins translation. That's still a difficult, still an open question, or many of the areas are still open. So Venki's lab is still working on it now, many years later.
But there was a difference in terms of what was known about the ribosome to what was known about APC, I assume.
Yeah, that's right. So when I started my PhD, I could basically read every single paper on the anaphase promoting complex. And it felt like a really unknown field, at least from a molecular perspective. There was so much to discover and very little known. With ribosomes, there was so much known already. Still lots to be discovered, but a lot known. And so when I started my own lab, I really felt like, I wanted to go back to that unknown where there was really less known molecularly about a pathway. And understanding how poly-A tails were added was just the right type of question. It was actually an old field. It had been discovered many decades before. We all knew that every mRNA had a poly-A tail. But really, how that works at a molecular level was completely unknown. And so it hadn't been reconstituted biochemically, the proteins hadn't been purified, really nothing was known. So I felt that was a good area to enter into.
When did you realise that was what you wanted to do as a group leader then?
That's a good question. When I was a postdoc, I used to have a folder on my desk, on my computer desktop that was called Interesting Complexes. Because I knew I wanted to work on a big multi-protein complex because I was interested in cryo-EM. And I felt like, I really wanted to identify the right area. So whenever I read a paper that was interesting, I would kind of store that PDF in that folder.
So what were the other things in there then, apart from mRNA and poly-A?
Well, so I think a lot of it did center around both RNA and E3 ligases, because those were kind of what I had worked on, and of course we're all very interested in. I knew the most about those fields. I remember I was really interested in cilia at the time too, which is also a very interesting topic, but I didn't go down that path.
Yeah. So it sounds like you were always pretty sure that this was what you wanted to do, to run your own lab and have a group, and is that correct?
I suppose from when I started my PhD, I was clear on that. But, I grew up in a small town. I had no idea what scientific research was. And in fact, I was the first person in my family to ever go to university even. So my goal was really to be a medical doctor. So I did my undergraduate degree in Canada. I applied to medical school. I got into medical school. But at that time, I was doing a final year project in a crystallography lab with Natalie Stranatka in Vancouver. And I loved it. I just loved the idea that you could see, the chemistry of biology. It was super cool. And I decided that's really what I wanted to do. And so I looked around for PhD positions. And then I think I never looked back. And I always felt like this research was amazing. And it's so much fun, and it's like a little playground for us.
So you didn't find it daunting then?
Well, I would say I did find it daunting. It was, it seemed like it would be hard to get there, but I never really doubted that I could do it, I guess. I just felt like if I was doing something that I wanted to do, that I was enjoying doing, I would keep on, moving forward. And so I think I always thought that I always just looked for the next opportunity. In a way, maybe I didn't have my eyes on the long-term goal. I didn't think about that too much. I kind of enjoyed the moment, tried to ask interesting questions, and have fun doing it. And each opportunity kind of came along as it did. Sure. So I don't think at that time I was thinking super long-term.
Yeah. Yeah, I think this idea of science as a career is actually quite modern. I mean, modern in the sense of probably in the time you've been a scientist, actually. But that idea of just seeing what came next and following your nose along a scientific path is a very traditional way to go, isn't it, as a scientist?
Yeah, I think I always used to say, well, we're never terminally differentiated, right? We can change what we do at any point.
Yeah.
And with that attitude, I think I always felt that, I would find a path and make it work.
Yeah. So you were a postdoc with Venki, and then you became a group leader here in 2009, is that right?
Yes.
So how did that transition happen then? Were you asked to apply or?
No, I think my life plan was always to move back to Canada. My husband's also Canadian, and I think we had always envisaged moving back. First, we imagined we'd move back after our PhDs and then after our postdocs. But actually, structural studies advertised 2 positions, group leader positions, and I thought, that sounds interesting. I would like to stay here. So I actually went to Venki's office and said, I noticed that there's some positions open. Do you think that you would consider an application from me if I applied? So I didn't hear about it from anybody. I just saw it being advertised. And in fact, I think I was on maternity leave at the time it was advertised. So I had to make a specific effort to come in and meet and ask the question.
Kind of surprising they didn't point out to you that there was a job going, but...
Well, I think I was clear that I was probably moving back to Canada, and so maybe it didn't really, occur to me or to them that it would be a suitable, appropriate opportunity.
Yeah. So you did all the job application process while you were on maternity leave?
Well, I was really lucky. My first daughter slept really well.
Okay.
She slept at night. And so my brain was active. Obviously, I wasn't in the lab, but I had 6 months off. And I felt like I was thinking about things, thinking about that folder on my desktop that called Interesting Complexes, and thinking about how I could develop that into a research proposal. And so I used that time to think and to think about what I would want to do and to basically craft my research proposal.
I think there's going to be a lot of new parents out there who will be extremely envious that you could think with a new baby, but...
I don't want to pretend that everyone could do that, and my second daughter definitely wouldn't have allowed me to do that, I don't think.
Oh, that's a relief then. So did they have any idea that you were interested in poly-A complexes before you put the job application in?
I don't think I had discussed it very openly. I think it was, that's something I came up with. And it was related, right? It was translational regulation, but also something else that I haven't mentioned is that removal of the poly-A tail. I also study removal of the poly-A tail. And one of the complexes that does that is another big E3 ubiquitin ligase. So there was a kind of connection between my postdoc and my PhD work conceptually.
In terms of mentors then, who would you say have been scientific mentors for you?
Well, so many. I think I've always, just talked to lots of people. I haven't felt any barriers in approaching scientists, especially, both at the Institute of Cancer Research and here at the LMB. I felt like people were approachable and they were all great mentors. So, of course, all of my supervisors were fantastic mentors. They were great. But I also had lots of other people I would ask questions of and discuss ideas with. So Daniela Rhodes' lab was just next to Venki's, and we talked a lot. We talked about science. We talked about families, having a family and being a scientist. And Kiyoshi Nagai's lab was also right next door, and we also talked a lot about science, and he used to ask me to help him review papers and stuff like that. So there wasn't real barriers between the labs. And, I felt like I could ask any of them questions, and they all did act as fantastic mentors.
So what do you think was your unique selling point then as a group leader? Why did they hire you?
Well, as I said, at that point, relatively few people were doing cryo-EM. It was still in its blobiology phase, so not routinely getting high-resolution structures, but still really valuable because we could look at these large complexes in their kind of more native state rather than taking them apart and looking at the individual pieces. And so my idea was really to do both of those things, right? To use cryo-EM to look at them in their state, in their native state, to use biochemical reconstitution to understand their activities, and then to use other structural methods to understand the bits and pieces and put it all back together and understand their mechanisms. And so from a technical perspective, I guess there was relatively few people that would integrate all of those things together.
I suppose Poly A was sufficiently different from everybody else who was working here as well that there wasn't a chance you were going to be competing with previous bosses. So.
Yeah, I think that's important too, right? I don't think that I would have been hired to work on the ribosome, for example. So it was sufficiently similar that I would be integrated into the department, into the division, but sufficiently different that I would have my own unique questions I was asking.
I'm intrigued by the fact that you say that you always just went to people and asked them questions. Did it never occur to you that they might be quite scary or rude or unpleasant or uninterested?
Well, I think at the LMB it's they're not, because especially the old LMB canteen was really crowded, and you'd walk upstairs, get your lunch and have your tray, be standing there, and there'd be only one seat left, beside some three other group leaders or something. So you'd have to sit down in the middle of them, and they'd make conversation. So that was just the way it was. There was no real hierarchy, and you didn't feel intimidated in the same way just to approach somebody. Yeah. Of course, I wouldn't want to ask a stupid question, and I'm worried about that, but I wouldn't feel intimidated to approach them.
So I want to take you back a little bit to when you started your lab, and I think that relates to what you brought with you today.
Yeah, so I started my lab in 2009. Around 2011, 2012, the new detectors for cryo-EM arrived at the LMB. And that was exactly the time when a postdoc, Chris Russo, joined my lab. And he is trained in physics. And he wanted to come to my lab, so he knew how to use the electron microscope as a physicist, and he wanted to learn how to use proteins in the microscope. And so he came to my lab, and these new detectors arrived. And so we decided, well, we can actually use those detectors to look in more detail at what actually happens when we're imaging the specimen. And it turns out that when you image it, the specimens move quite a lot. And so while Chris was in my lab, he decided that this was something that we could resolve. We could try and fix it, make better specimen holders, and use those for better imaging. And he did that, and we developed these. This is some glass fusing where there's some gold grids in here. So his new supports were made of all gold. And those supports then almost eliminated the specimen movement in the microscope. So again, kind of, one of those abilities in the LMB to kind of fix the methods or develop new methods when there's a gap. And so a friend of mine, Jude Short, who also works here at the LMB, does glass fusing as a hobby. And so she took some of our very first grids that we made and put them into glass. It was a really fun time because Chris and I did a lot of imaging on the microscope at that time, it was before we had real automated imaging, so we had to manually collect all of our data, and we would do shift data collection all night on the microscope. But it worked out in the end, and there's some of the first grids.
Rather lovely, isn't it? So I take it those grids are empty. There's nothing
on them anymore.
So apart from the transition from blobology to wonderful resolution, how has EM changed over the time you've been involved in it? You were saying manual collection has obviously gone away.
Yeah, that's right. So it's become much easier to use. So, we have hundreds of users on the microscope now compared to the 7 registered users back in 2011. And you can use it now basically as a user instead of being an expert. And that has really opened the door to so many more projects. So that's one major change. Of course, as I've just mentioned, the other major change is that we can get much higher resolution much more routinely. And that's because of better detectors and better software, better instrumentation. So they all combined to really allow us to get make atomic models of our proteins from our EM maps.
Is that changing your ambitions for the years to come?
Well, it already changed them, I would say. We knew that EM had the potential to get to this resolution, but it happened really quickly, actually. Over a space of, just a few years, we went from, blobs to atoms, basically, or atomic models. And so it changed the way I do experiments, but the way everybody does experiments. So now many labs at the LMB use cryo-EM in their research. And, we can think about doing things like looking at reactions as they happen, or looking at time-resolved processes, or looking at our protein complexes within the cell. So I think there are so many more questions. It's opened the door to so many more questions.
Have you still got an interesting complexes folder?
I don't have that folder anymore.
Yeah. You're happy with the complex you've got?
I think there's always diversions off our current projects. Interesting diversions off of them. So yeah, there's lots of interesting ones. I don't have the folder though. Yeah.
So you're with Poly A for the duration, you think?
For now, yeah. Absolutely.
Sure.
Yeah.
Oh, right.
Yeah. There's lots to do because we're not only studying how it's added, but how it's removed. So there's quite a bit of biology there.
What are the downstream implications of knowing how that happens then?
Well, mRNA has become very topical, and the mRNA stability and how efficiently it's translated has become very topical in the last few years because of mRNA therapeutics and vaccines. So, downstream, understanding how the poly-A tail influences mRNA stability and translation is super important for mRNA therapeutics. But it also turns out that some of this machinery is deregulated in lots of diseases. And a few years ago, we were involved in a collaboration with Novartis, who found that one of their drugs, for which they didn't know the target, targeted the polyadenylation machinery. And so that was really effective in treating, they screened cancer cells, and it was effective in a bunch of like Ewing sarcoma and AML and a bunch of different types of cancer. So targeting the polyadenylation machinery could turn out to be a really interesting avenue.
Is that something you're interested in pursuing in your own lab, or will you leave that to other people?
I think it is definitely an interesting thing to do. I mean, we've now been able to reconstitute the entire pathway in vitro from recombinant proteins. So I think we have all the tools that we would need to do in order to start thinking about drug discovery. And those, of course, would be useful for disease, but also for as tool compounds to understand the pathway in vitro and in cells. Yeah. So that's something that we're thinking of.
Now I want to switch tracks entirely and talk to you a bit about being a woman in science. So there's an awful lot of negative stuff said about women in science and how difficult it is, but you seem to be a positive exponent of this. Why is it that you think being a woman in science is a good thing?
Well, first of all, being a scientist is fantastic because we get to do fun things every single day. So, and if you love your career, love what you're doing, I think it's easy to make things work. So, yeah, it's important that, first of all, if you're a scientist, you love doing it because, I sometimes, wake up thinking about an experiment because I love it, not because, maybe because I'm obsessed with it. Yeah, maybe. But in terms of being a woman in science, I mean, actually, scientists are super diverse. You can be who you want to be as a scientist. And we also have really flexible careers, flexible hours, flexible working patterns. And that's super great when you have a family to have that flexibility. So I think there's really a lot going for it that, means it is a fantastic career to be as a woman. Yeah. I never questioned whether it would be possible to have a family, and
why do you think that is?
Why I never questioned it? Yeah. I guess my parents both worked full time. I didn't see that there was any real barrier to having a career and being a parent, and so I just went for it. It didn't occur to me that it wouldn't be possible. So yeah.
Do you think there are people still being put off continuing who are good enough to have come here but are still being put off continuing because they worry about career versus family?
Yeah, definitely. And there are some really hard times, right? When children are young, and sometimes it's hard to know, like, where do you put your time and efforts? And sometimes you feel like, or I felt like I'm not spending enough time with my children, so I'm not a good mom. I'm not spending enough time in the lab, so I'm not a good scientist. And you just have to kind of compartmentalize your time as much as you can. And I would set myself goals. In the lab, I would set myself specific times that I would turn off from the lab and be with the family. When my older daughter was in nursery, we didn't have a nursery place for her for 2 mornings a week. And so very reluctantly I said, okay, I'll take 1 morning off if my husband would take 1 morning off and we would look after her. It was the best decision I ever made, No one noticed that I didn't even come in for 1 morning a week. And it was really special time with her.
There are very few jobs that would allow you to do that, I guess, at the time where you were doing that. Maybe now it's easier, but yeah.
Or run home because one of them broke their glasses and I need to deliver a new pair to the school, or attend a concert in the middle of the afternoon. So all of those things were easy to do. In part, I made sure that I lived close to the lab so that it was easy to get back and forth.
And I imagine you're very good at saying no to things that aren't vital.
That's a hard thing to do. You do have to say no. And my strategy is to say no right away. Not to think about it for too long, because then I'll agonize and think, maybe I could manage it, maybe not. I just say no right away. If I haven't said no right away to you, then I'm very seriously considering it.
I'm glad you didn't say no to this interview. I guess a lot of the things that will make doing science easier is that there are big changes in research culture that are happening now. And you're a chair of a Wellcome Grants panel, I guess. Is that right?
I am, yes.
And therefore are having to implement some of these changes. How is that working out?
So I actually just got a Wellcome Grant as well. And so I know from an application perspective, the Wellcome is considering research culture, and that's part of the grant application. And I think more and more funders and employers are thinking more about it. So what do I do as a researcher to promote positive research culture? And that's important because, it values not only my science, but also how I behave in the lab, how I mentor people, how I interact and nurture their careers, whether that be in a science research career or an alternative, I don't want to say alternative, a different scientific-based career or a non-scientific career. And so I think that's going to change how people experience science, because this is now becoming much more ingrained and much more important that, we can't just be successful scientists. We have to be good mentors. We have to have a good environment in our lab as well. And I hope I always had that, but, now it's actually part of the evaluation criteria. So I think that's a really positive move.
Yeah. So things are looking up for scientists of the future then, you think?
I think so, yeah. I think it will make everybody think more carefully about what they do to promote that, teamwork and, career progression and integrity in the lab.
Yeah. And on that note, thank you very much for coming to talk to us, Lori. It's been really interesting.
Thank you, Kathy.
I was having trouble digging around in my head to phrase things